Hello, my name is Adrian. I'm a PhD student at Iconeus and Physics for Medicine Laboratory. And I work on the development of new modalities dedicated to 3D functional ultrasound imaging. So, functional ultrasound allows to map the brain activity thanks to neurovascular coupling effects. And most of studies are performed in 2D using linear arrays in awake and anesthetized rodents. But it's limited to one plane, and functional connectivity networks are organized in 3D. So this is why we need to develop new probes to image the whole volume. Matrix transducers could be used, but they are not enough sensitive to go through the skull. And linear arrays are highly sensitive, can go through the skull, and we could motorize them, but we are limited to 4 planes, which is not enough to cover the whole brain while maintaining a good temporal resolution. So this is why in the last year I developed with Iconeus and Physics for Medicine Lab a new approach, the multi-array probe, which is patented by Iconeus and which is based on the same acoustics as linear arrays, so highly sensitive. And there is 4 linear arrays which allow to insonicate 4 planes and to record 4 slices simultaneously. And actually it allows to map with 16 slices at 0.4 Hertz the whole brain, so we can reconstruct the whole vasculature, and it's sensitive enough to do this imaging through the skull. And so to validate this approach we did experiments, and so we started with resting state functional connectivity, and we computed the functional connectivity matrix with 372 volumetric Allen regions after registration. And as you can see, we found strong functional connectivity patterns common to different animals in the cortex, in the hippocampus. And we also did seed-based analysis. So here it's an awake mice, and as you can see, we have strong functional connectivity that spreads through more than 2 millimeters in the brain, so in 3D. And finally, we also tried visual system stimulation using a white flash. And as you can see, in one single experiment, we could measure a strong activation in the 3 main visual system regions, so the LGN, the superior colliculus, and the visual cortex. So it shows that this probe is sensitive enough to go through the skull and to record the functional connectivity in the whole brain. So in terms of field of view, temporal resolution, and sensitivity, the motorized multi-array is the only solution that allows whole brain transcranial mouse connectivity studies.